Welcome to the second video on sex chromosomes and gene linkage. In this video, we will discuss the concept of gene linkage and how to determine if genes are linked. Previously, you learned that in a normal dihybrid cross, the two genes do not have to be inherited together. This was Mendel's law of independent assortment and occurs because, as seen in this figure, homologous chromosomes can align in any orientation during metaphase 1. For example, the dominant A allele could align on the same side of the cell as the dominant B allele, resulting in gametes of dominant A, dominant B, or recessive A and recessive B. However, there was an equal probability of the recessive B lining up on the same side as the dominant A. This would have provided gametes containing dominant A with recessive B and recessive A with dominant B. This provides an equal probability of obtaining any of the 4 possible gamete combinations. However, Mendel was only able to observe this pattern because the traits he was studying were controlled by unlinked genes, or genes that are on separate chromosomes. We will now add a third gene, gene D, to this figure. The dominant D allele is on the same chromosome as the dominant A allele, and the recessive D allele is on the same chromosome as the recessive A allele. When genes are close together on the same chromosome, such as gene A and gene D, they are called linked genes, and will no longer follow the same 1-to-1-to-1-to-1 pattern we see in independent assortment. In this case, the dominant D allele is physically attached to the dominant A allele, since they reside on the same chromosome. Whichever gamete contains that chromosome will contain both the dominant A and the dominant D alleles. The same is true for the gametes that gain the other chromosome. They will now contain the recessive A and recessive D alleles. As we see in the resulting gametes, there are only 2 possible combinations observed when we look at the A and the D genes, with a 50-50 chance of obtaining one or the other. The fact that the genes A and D no longer show independent assortment indicates that they show genetic linkage and are on the same chromosome. While linked genes are often inherited together because of the physical linkage of the chromosome, it is possible to separate linked genes through crossing over. As discussed previously, crossing over occurs in prophase 1 of meiosis and is a physical exchange of chromosome parts. In the example here, A and B are linked genes, with the dominant alleles associated on one chromosome and the recessive alleles on another. However, when crossing over occurs between them, the linkage is broken, and new chromatids are generated that now contain the dominant A allele with the recessive B allele, and vice versa. The chromosomes with the original allele combinations are called the non-recombinant or parental chromosomes, because they will match the phenotype combination that was observed in the original parents. The chromosomes resulting from crossing over are called the recombinant chromosomes. Crossing over results in 2 recombinant and 2 non-recombinant chromosomes. This is because crossing over occurs at the 4-strand stage of meiosis, when each homologous chromosome is a pair of sister chromatids, but only 2 of the 4 strands, 1 sister chromatid from each homologous chromosome, are included in any crossover. The frequency of recombination is a measure of the distance between linked genes. Let's examine these 2 chromosomes with a variety of genes along them. Note that only 1 of the sister chromatids is shown for simplicity. For 2 genes found on the same chromosome, they may be located very far from each other, such as the A and the R genes. In this case, crossing over is likely to occur because it could occur anywhere along this chromosome and it would lead to the separation of the A and the R alleles. In fact, genes that are very far apart, such as A and R, are often considered unlinked because the alleles are typically separated during crossing over. Alternatively, the genes may be found very close together, such as the T and the B genes. This makes a crossing over event occurring in between them much less likely, because the only place it could occur to separate the two alleles would be here between the genes. This means that the frequency of recombination between any two genes ranges from 0% when crossing over never takes place to 50% when the genes are so far apart that crossing over between them will take place. The less recombination between the genes, the closer they are on the chromosome. Because the frequency of recombination can be used as a measure of the physical distance between genes, it can be used to make a map of the genes on the chromosome. This is called a genetic map and diagrams the relative position of genes along the chromosome. The units in a genetic map are called map units, and one map unit is equivalent to 1% of recombination. In order to make a genetic map, you must first determine if two genes are linked, and then determine what the percentage of recombination is that occurs between them. To do this, you perform a specific type of genetic cross. First, you take two true breeding dihybrids for the traits of interest and mate them to generate a heterozygous F1 offspring. In this example, the parental cross is represented by a homozygous red tall plant with a homozygous white short plant. The heterozygous offspring are formed because the plant inherits one chromosome from mom and one chromosome from dad. You then complete an F1 test cross where the dihybrid is crossed to a homozygous recessive individual. The offspring from this cross will determine if the genes for color and height are linked by determining if there was the standard 1:1 to 1:1 ratio of phenotypes observed in the F2. Remember, a departure from that ratio identifies gene linkage, as seen here. All of the F2 offspring will receive a recessive A and recessive B from the test cross parent. This is important because it means that the phenotype will be determined by the heterozygous parent. In the heterozygous parent, if no crossing over occurs, most of the time the F2 offspring will inherit either the two dominant alleles, making them red and tall, or the two recessive alleles, making them white and short. However, if crossing over did occur in between the A and the B genes, then the offspring would have inherited the recessive A allele along with the dominant B allele. And the dominant A allele would now be with the recessive B allele. We will now examine the phenotypes and numbers of the F2 offspring. In this cross, we observed 350 tall red plants, 325 short white plants, 40 tall white plants, and 35 short red plants. This is clearly not a 1:1 to 1:1 ratio, indicating that the color and height genes are linked. The last step will be to determine how far apart the linked genes are so that you can make a genetic map. The distance between the genes is equivalent to the frequency of recombination. In order to determine the frequency of recombination, you must first identify the plants that match the parental phenotypes, and those that were recombinant phenotypes. The original parental generation of the cross on the last slide were red and tall, crossed with white and short. Thus the red tall and white short offspring are the parental phenotypes, making the tall white and red short the recombinants. The frequency of recombination is then the number of recombinants divided by the total number of offspring. In this example, that would be 40 plus 35 divided by the sum of all of the plants total. This is equivalent to 0.1 or 10%. This means that when you draw the genetic map, Gene A and gene B are 10 map units away from each other.